The Amazon rainforest contains an incredible diversity of tree species. Many estimates are that there are more than 10,000. Some of these species are very important, both because of their role within larger ecosystems, as well as for direct human uses. Our research focuses on better understanding genetic differences among tree species related to the Brazil nut, because of this group's economic and cultural importance, stemming from the use of its fruits, seeds, and wood, as well as their prominence in Amazonian forests. In many areas of the Amazon, these trees are among the most abundant species. They provide food and timber resources for human use and occupy a role within the complex ecological fabric of the Amazon rainforest. Our project is taking place within a larger effort to understand how demographic differences among tree species might contribute to differences we observe in the distribution of species across the landscape. The work for this project is focused on a forest plot in northern Brazil near the city of Manaus, which is managed through an expansive program called the Biological Dynamics of Forest Fragmentations Project at Brazil's National Institute for Amazonian Research. Because we are confident in the species identification of trees within this permanent plot, this is a great system to build our understanding of genetic differences between species that can be very difficult to distinguish from one another based only on their visible characteristics. To monitor the long-term dynamics of these species, each individual must be identified and tagged in order to make sure we can identify the same tree again later. Then, the tree's location within the plot is recorded, along with its diameter and a few other details. Collecting genetic material, like DNA, from leaves while we're in the field allows us to identify what makes these species genetically distinct. It also allows us to create a reference database of DNA to identify unknown trees in the future. This type of information is likely to become even more important as we are forced to make decisions about how to allocate limited resources in a future of changing climate and expanding economic pressures. Each collection we make is accompanied by a pressed herbarium voucher, which ensures that the genetic data we generate is always tied to a physical, verifiable specimen, and that our results are repeatable. As technologies improve and new methods are discovered, we continue to find new uses for these collections, meaning that they have the potential to contribute to new scientific knowledge well after our project ends. We work with our collaborators to ensure that a duplicate of each sample we collect remains at our host institution, where they will be available to be used in future projects and in new ways. We are also able to incorporate archival samples into our project to expand the scope of our research beyond what we could accomplish otherwise. By using these collections to generate new knowledge and expertise, we increase their value, and everyone involved benefits from the exchange. Through the many facets of this work, we hope to better characterize the genetic diversity of Amazonian trees in order to help make informed decisions about how the world's tropical forest resources are managed and studied well into the future.